Practically in this webinar, we want to introduce quickly a preliminary overview about the enzymes. The enzymes are important tools for the application, both in research, but mainly in the industrial processes. And INCITE is a concrete example. In fact, in INCITE project, we have the final target to prepare two demo plants based on chemoenzymatic processes where the enzymes play a strategic role. In these slides, we'll meet progressively several points, several items, starting from the main characteristics of enzymes. And obviously, when I fix what is the right enzyme for me, what is the right source, the starting material, what is the right starting material to have the right amount of enzyme useful for my application. The downstream step, because generally in the process you can recognize two blocks, two processes, the upstream process and the downstream process. The upstream is focused to the cells and the cell cultivation, and the downstream is focused on the purification, the extraction and purification of my biocatalyst. Then we move to the biocatalysis, is the real use of enzymes for the production of my product of interest, with or without the mobilization of enzymes. And obviously, we'll have a link of all concept in the INCITE project to understand what we have considered of all world of enzymes for our project. We start obviously from the introduction of enzyme. The first step is what is an enzyme? Well, simply speaking, enzymes are proteins, practically. Proteins, and the structure is composed by our biopolymers. Biopolymers composed by single monomers. These monomers are represented by amino acids. Practically is a long chain, is a polypeptide chain composed by several amino acids. The sequence of amino acids is very important because we have a specific sequence of amino acid for each proteins or enzymes. Obviously, every kind of proteins have a particular role. What is the role of enzymes? Enzymes are fundamentally catalysts. In the cell, all metabolic processes are mediated and catalyzed by enzymes. In fact, enzymes are catalysts. We can accelerate a reaction, transforming a substrate to a product. At the end of the reaction, as enzyme is a catalyst, must remain inalterated. Is a typical property of every catalyst, biocatalyst or chemical catalyst, but at the end of the reaction, the catalyst remains the same. The structure of a protein, and consequently of an enzyme, is quite complicated because it's a very long, a very articulate structure. In fact, when we discuss about the structure of a protein, we recognize more or less 4 levels of structure: the primary, the secondary, the tertiary, and also the quaternary structure. What intended with this one? Practically, the primary structure is the simplest. With the primary structure, we considered the sequence, the amino acid sequence of the protein. Practically, I've all the sequence and the name of the protein, starting from the N terminal up to the carboxylic terminal. N and C terminal. And in the middle, you have all the sequence of amino acids. But obviously, the protein are not a linear structure. There is the secondary structure. The secondary structure is the beginning of the complexity of the protein. practically for each amino acid, we have an interaction with the other. There is an interaction to each other, and the consequence is a more complicated structure. In fact, with secondary structure, we consider some particular structure in terms of helicoidal or plane structure called alpha helix or beta sheet. And we start in the secondary structure also with the first interaction between the amino acids based mainly on hydrogen bonds. Then we enter in the tertiary structure, the best of the structure, because in this case, the protein in the refolding can form a specific disulfide bridge. Practically two residues of cysteine can link to each other, and this bridge can consolidate the structure of the protein. And then we have a lot of interaction of different natures, ionic, hydrophobic, van der Waals strength. In this way, you have at the end a three-dimensional configuration of the protein, very complicated with a lot of interaction, and practically the final native structure of the protein is a three-dimensional configuration with a lot of interaction, and the protein have the final structure with the lowest energy state. Obviously, is a flexible structure, because if I change the condition, the environment where the enzyme is, for instance, a solution, a buffer solution, if I change the buffer, the pH value, the temperature, the structure is flexible and can modify also its shape. Another concept very important for enzyme is the active site. What is the active site? That is, active site can be defined such as a small area in the structure, a sort of pocket on the surface of the protein, where are located inside this pocket, in this site, are allocated few amino acid residues with a particular configuration and position. And the active site is important because is the specific area where the catalytic action is performed. Practically, in an active site, the substrate or a part of the substrate enter in the active site. There is a specific interaction with the amino acids allocated inside, and at the end, this interaction facilitates the reaction, the catalytic action, forming from the substrate the product. Obviously, in this reaction, this particular catalytic reaction can participate not only the active site and the substrate, but also several cofactors. Depends enzyme per enzyme. And I can also involve other molecules. We can facilitate the reaction. Metallic ions, cofactors such as thiamine, pyridoxal phosphate, and so on. Depends. Another point important in the characterization, and okay, I've got my enzymes, but can I measure, quantify my enzyme? Yes, generally, considering, for instance, a chemical approach, we have generally a direct estimation. Imagine an organic molecule. I want to know the concentration. I use gram per liter, milligram per ml, percentage, moles. With the enzyme is different. Okay, theoretically, I can use the same approach, but only if I've got a very purified enzymes. But generally, I've got a mixture of protein where there is also my enzyme of interest. In this case, we use an indirect approach to estimate enzymes. Practically, we measure the specific catalytic capacity. This catalytic capacity is called activity. That's why we call usually of enzyme activity, to know how many enzymes I've got and if it can work. Practically, the activity have a measured unit and is called unit. Very simple to remember also. What is the unit of enzymes to determine the activity? The unit is defined such as the amount of enzymes we catalyze the transformation of 1 micromole of substrate per minute under standard conditions. The standard conditions are the conditions that are developed when I use a specific analytical method to estimate the activity of enzymes. If we consider the international system of units, practical unit is not included. But the international system use the CATAL. The CATAL is another unit to measure the activity of enzymes, the official measure, and is intended such as the quantity of enzymes we can produce 1 mole of substrate per second. Is a very big system. So generally, every day, and in the all labs and plant, we generally use the unit. So we can have the estimation of enzymes assaying the activity, and the activity is expressed in unit, and generally this unit are used instead of CATAL, is more practical. We like to remember also the specific activity. What is the specific activity? Specific activity is a ratio. It's a ratio between the units, the enzyme activity, and the milligram of total protein that I've got inside my solution or my mixture of enzymes. Because it's useful. Why is useful? It's useful because it's important during an enzyme purification. Because if I consider several steps to purify my enzyme, I've got a purification index which increases step by step. When I determine the specific activity, I've got an increasing of specific activity expressed in units per milligram. I've got a measure of my purification, because when increase the specific activity, I'm reducing the total proteins maintaining my enzyme. Another phenomenon that we have to remember when we work with enzyme is the inhibition. Enzyme inhibition is a common phenomenon. And what do you mean with inhibition? Well, practical inhibition is in interaction the enzyme with a different molecule able to reduce drastically or to block completely the activity of the enzymes. We can recognize 2 kinds of enzyme inhibition: reversible or irreversible inhibition. And then we can divide also the reversible inhibition in 2 categories: the competitive or non-competitive inhibition. To understand better, we have 2 simple examples. On the left, an example of irreversible inhibition. Is irreversible because in this case, we have got a covalent bond between the enzymes and the molecule, the inhibitor. In this example, is a scheme very simple, but in this case, we have got the enzymes with a residue highlighted, is a serine residue, and is in contact with an organic phosphoorganic compound. In this case, I've got a covalent bond between the phosphoorganic compound and the serine. In this case, is completely inhibited. because I've got a covalent bond, it's very stable. It's not possible to have a reversible effect because it's very stable. In particular, what is this example? Enzymes, sorry. Enzymes is in this case acetylcholinesterase. Is an important enzyme for the neurotransmission. And this kind of phosphoorganic is a nerve gas. Practically, this is the mechanism of the nerve gases. Nerve gases are recognized such as chemical weapons, are mass destruction weapons, very dangerous. And when a person is killed by means nerve gas, the mechanism is this one. Practically, is blocked completely the acetylcholinesterase. Different the reversible. The reversible, I can come back to the initial situation. In the competitive inhibition, what happened? Practically, the inhibitor have a structure quite similar to the substrate. Practically, is a competitor, can enter in active site instead of the substrate. In this way, for competition, can reduce the access of the substrate in the site, in the active site, and then I've got a strong decreasing of the activity. In the non-competitive case, instead, I've got always an interaction between the inhibitor and the enzymes, but not in the active site, but in another part of the structure of the protein. This interaction causes a deformation, a modification of the structure, which can influence the interaction between active site and substrate. Also, in this case, I got a decreasing of the activity. It's important to know the inhibition because I can use this inhibition such as a tool if I want to inhibit, for instance, a contaminant, another enzyme that is not useful for us. Or in some cases, it's important to know if I've got some potential inhibition because I can lose my activity without understanding what is the reason. In this case, if I got a potential inhibitor, it is important to eliminate it by purification or other approaches. When I decide what is my right enzymes, the first question and the first problem that we meet is what is the right source where I can find my enzymes in with right quantities and a source that is always available. Practically for industrial application for enzymatic process, as enzymes have a biological origin, all source originated from a biological origin are suitable. Practically, we can use material from animal origin, plant origin, of microbial origin. For instance, also on the market, if you think about some examples, imagine, for instance, two enzymes such as pepsin and trypsin. Often these are enzymes originated from an animal. But in our case, for this webinar, and in general for industrial application, the most important source for enzyme are the microorganisms. Why? Because microorganisms are extremely diversified. We have really a world of microorganisms. We have thousands and thousands of microorganisms, and I've got an unlimited number of enzymes available. And then we have got also several and important advantages. The variability, the versatility, and also the availability of these microorganisms the possibility to have a scale up completely independent from environment, seasons, and so on. Imagine, for instance, the plant in terms of source. I have a lot of biomass. I use a lot of land. I have to consider the seasons. It's more complicated. Instead, in this case, it's simpler, the use and the scale up. And then, thanks to the evolution of all techniques of molecular biology and genetic modification, we can use the cells in terms of host cell for the heterologous expression. So, summarizing, I've got a lot of microorganisms, and if you enter in the web, you can look for several strain collection, for instance, ATCC in the States, DSMZ in Germany, and so on, where it's possible to buy with a cheap cost a lot of kind of microorganisms. Obviously, they are wild type, the original microorganism, and to use in an industrial application is important the development of the strain to have right biomass and enzyme quantity. Is quite convenient the scale up, as we have anticipated, because the microorganisms are independent from the environment, but they are also very fast. Imagine, for instance, Escherichia coli, E. coli. E. coli is often used and is used as also from our company for the INCITE project. Every 20 minutes, we have a new cell. Practically, you start from a single cell. After 20 minutes, we have 2 cells. After other 20 minutes, 4 cells. Practically, you have got an exponential growth. And if you consider a cell cultivation with a duration of 1 day, after 1 day, per liter of broth, you can obtain tens or hundreds grams of cells. Practically the biomass that we'll use for the extraction of the enzyme. It's important then the heterologous expression. Practically, a particular enzymes of a particular microorganism or animal or plant is expressed inside a different cell, the host cell, after a modification, genetic modification, to improve the performance. This activity has been done, for instance, in INCITE project, because we used a wild type esterase, and before the starting the INCITE project, we prepared the right enzymes, manipulating the sequence, the DNA, and consequently the protein, and then express the video enzyme inside in E. coli in terms of host cell. The first part is, okay, I've got my source, my enzymes, I have to produce it. The first approach is the upstream part. The upstream is the biomass production. A lot of cells, because I need a lot of cells to produce my enzymes. The biomass production is performed in a particular reactor called generally fermenters. This is a picture of a fermenter. It's not casual, but this is the real fermenter that we'll use in INCITE project. On the left, you have the main controller. In the middle, you have the vessel of the fermenter. Practically, is a reactor. It's a reactor with a stirrer, a jacket, the control of pH, temperature, quantity of water, oxygen, and so on. And practically, what is the main characteristics? Yes, it's a reactor similar to the other chemical reactor, but in this case, we work under sterile condition because it's important to maintain my cells without contaminant. At the end, I obtain the biomass inside the liquid composed by the broth, which at the beginning contain all components necessary for the cell growth. At the end, I have to decide what is the part that is of my interest, because the enzymes can be intracellular or extracellular. In some cases, in the best, the enzymes is inside the cells, but in some cases, the cell there is the secretion in practically of the enzymes in the environment, in the liquid, in the broth. Is an extracellular enzymes. For instance, imagine a very different process. Imagine the chymosin. Chymosin is the most important enzyme for the dairy industry. The chymosin produced by fermentation is an extracellular enzymes. In this case, the part of my interest is not represented by the biomass, but by the exhausted broth. I have to know exactly what is the part of interest. Remembering is always the INCITE project, we have a cultivation of E. coli, okay, a transformed E. coli. E. coli is not capable to secrete the enzymes. Is not E. coli doesn't have this property. So the enzyme is inside. In this case, I have to harvest the biomass and then continue with the downstream part. The downstream part where, starting from my biomass, I can think about the extraction of the enzymes and to have at the end not the biomass, but a clear solution containing my enzymes. As the enzyme is inside the cell, I have to break the cells. I have two categories of methods, non-mechanical or mechanical method. In the first case, we have some methods used in general on small scale, but we can repeat also on the large scale. We can use our physical approach, using, for instance, heat or freezing, or I can use an approach adding an agent such as enzymatic or chemical agent. For instance, lytic enzymes such as lysozyme, mannase, glucanase, detergents, organic solvents. In this case, I can obtain good results, but I have to consider that are adding a new component in my mixture, in my solution. And probably I have to think about at the end if I have to remove this component or not. It's more comfortable than mechanical methods, because in this case, we have the ultrasounds, but mainly on small scale, but for the approach in industry are very important, the ball mill and homogenizer. Ball mill, what are? Our mill practically is an equipment with a particular chamber. The lysis chamber is practically a sort of small reactor when you have a vorticus steering with the presence of glass balls, glass beads. Practically feeding your cell suspension with the agitation, with the steering and the interaction with the glass beads, we have the disruption of the cells. And at the end, we obtain the lysate, practically a suspension where we have got all proteins in the solution and all the cellular debris. The homogenizers are different. The picture, for instance, is an example of homogenizer, is a press, practically. The cube is the engine, and in front you have the chamber and the valves able to increase the pressure to my sample. Practically, I can feed in continuous my cell suspension, They are submitted to a high pressure, at least 800 of bars. Then we reduce drastically the pressure, and passing from the high pressure to atmospheric pressure, I got the salt disruption. In this case, it's more comfortable because I don't have glass beads and so on. In INCITE process, we use this one. At the end, we have got a D8. What is my problem now? And what is my problem before the disruption? The separation. Because we have practically in the process, the first step is always a separation. At the beginning, you have to separate the salts from the broth. The cells harvested are suspended in a buffer solution and then is desated. But at the end of cell disruption, I have to clarify my solution. I can use this suspension with a lot of salt debris. No, I have to separate the solid part from the clarified solution. These steps can be performed adopting two techniques, the centrifugation or the filtration. We can't enter in detail, obviously. The name that we have listed are a typical name of a kind of centrifuges used in industrial application. But in general, we can use centrifuge of several kinds, tubular disc, chamber. But in general, a centrifuge is an equipment with different chambers inside to facilitate the sedimentation of the solid, adopting a centrifugation, an increasing of the centrifuge force. Practically is a physical separation. Instead, if I use a filtration, and in industrial often use the tangential filtration, what is the meaning? Practically, I've got a filter, and my solution of suspension or cell, lysate or odor move parallel on the filter and not orthogonal, because in this way I can avoid the thin layer and the strict reduction on the flow. Also in this case, I have different kind of filtration, flat, spiral, hollow fiber. In the picture, this is the real unit that we use in INCITE. Practically, on the left, in the middle, there is the pump, the controller on the right. On the left, you can have a look to an older device of filtration. In this case, we have got flat membranes called cassettes. If you have a look to the stainless steel structure, in the middle, you can find the cassette are yellow, practically. And these are the filters. And in this way, you can clarify it completely, the lysate obtained after homogenizer. At the end, at last, I've got my clarified solution. At this point, I have to decide. But this solution is suitable and enough for my application, or I need a further purification. In the second case, I can choose several steps of purification if I need a stronger purification. What is okay? I can't also in this case enter in detail, but we have first we mention one of the best application techniques to purify an enzyme. This is the chromatography. What is the chromatography? It's a particular technique, very consolidated, but in general is performed in column, glass column or stainless steel column for the large scale, where I can recognize the two phases, one liquid phase, the mobile phase, and one fixed phase, the stationary phase, composed generally by beads of resin. practically my mixture, my solution with several kinds of proteins in order is eluted along the stationary phase that composed a bed inside the column. Passing through the stationary phase, the solution have an interaction between mobile phase and stationary phase. The affinity between the two phases determinate the separation of several compounds. In some cases, some proteins have a strong interaction with stationary phase, and they are absorbed or slowed down during dilution. In this way, I can separate several compounds. In biotechnology, I can use all kinds of chromatographies because I've got several kind. What is the difference? Is the principle adopted for the separation because are available a lot of stationary phases, particle resins, where this polymer has been modified. Adding some functional group, we can supply a particular properties to the polymer. For instance, in the ion exchange chromatography, I've got some charged group on the resin. There is an ionic interaction with the protein. Or I can use a functional group with an hydrophobic structure. Some proteins, such as lipases, for instance, have some part of the structure with a high number of hydrophobic amino acids, which can interact with this group. It's an hydrophobic interaction. In the gel filtration, the separation is based on the different molecular weight of the protein and the affinity that is the best for the biotech. In the affinity, I've got a specific interaction between a chemical group linked on the stationary phase and my molecule. For instance, imagine when I've got an antigen fixed on the stationary phase and the antibodies to purify the antibodies, for instance, or when I use proteins with in the structure a short tail of histidine. The amino acid histidine have a strong interaction with a metallic ion. In some cases, I can use on the surface the metallic ion for the interaction with this his tag, and I can separate my proteins with his tag from the others. So, summarizing, I've got a lot of possibilities, but I have to decide what is the target. Is necessary or not necessary, the chromatography? In this slide, this is a very simple scheme where is summarized all possible steps. This is a real process. It's a process to purify a biosimilar, in this case, somatropin. Is covered where I have to cover the details because it's not possible to tell really the name. But this is a typical upstream and downstream process. At last, we can get to the final approach, the biocatalysis, but I can use the enzymes with several formulations. I can use directly the solution, the purified enzymes. I can transform the liquid enzyme in a solid enzyme, say, limited in the water. I can use the cells, but these are all biocatalysts. But I can use a further approach, the immobilization. I can join my enzyme to a carrier, practically is an example in the picture. with the electronic microscopy is these are some bills resin. We can use it for the immobilization. Practically, with a reversible or reversible bond, I can link my enzymes to a solid carrier, and at the end, blocking the enzymes on the resin, I can use it in terms of solid enzymes. What is the advantages? Best, practically, I can use these enzymes in an heterogenic catalysis because I've got a solid and a liquid. It's very easy, the separation. I can control better the reaction. I can reuse the enzymes for many cycles. And in many cases, I can obtain also a best stability of enzymes. In the biocatalysis, I can use all these biocatalysts. And the biocatalysis biotransformation, what is? It's the use, generally, of living organisms or part of them, such as the enzymes, to obtain a relevant amount of compounds of interest. It is important using enzymes. Their selectivity is the key of success of enzymes. In fact, we can recognize the three levels of selectivity in enzymes. The chemoselectivity, when the enzyme is able to recognize in the molecule, the substrate, a particular group. Also, if you have present also another group with a similar reactive level. For instance, imagine if you have got an ester and an amide. The reactivity is similar, but the enzyme can recognize the one or the other, ignoring one or the other. It's very important because in chemistry is very difficult, this step. Instead, with enzymes, it's very easy. Also, the regioselectivity. Imagine to have a molecule with two esters. The enzymes, if possess this selectivity, can recognize only one of the ester, ignoring the other. And the best is the stereoselectivity, where the enzyme can recognize in the rachemic mixture only one enantiomer. Practically, the enantiomeric isomers are molecules with the same structure, the same composition, but a different arrangement of the atoms in the space. It's very difficult chemically to discriminate this one. Instead, the enzymes can be perform this selectivity. We can conclude with the approach in enzyme of the enzymes in INCITE project. In INCITE, we can recognize two processes. One oleochemical part and one agrochemical part. In the oleochemical part, in both, we use a biocatalyst. In the first, we use a commercially mobilized enzymes for a selective esterification of the substance. Instead, the part where the company that I represent is involved is the agrochemical part, where we have got a not commercial enzymes because it's not available. We have, with the research, we have modified, improved the enzymes and esterase in this case, with a very high selectivity, stereoselectivity, because the particularity is that in this reaction, we have got a racemic mixture where we have got 4 different enantiomers, but only one is suitable for the reaction, because as the unique, we can at the end produce an active compound. The others are inactive. The enzymes with esterase is able to recognize one, ignoring completely the others. It's very strong. And in fact, we about this one. In this slide is summarized the process practically. For this esterase, it's not necessary chromatography steps, but we have only the part of upstream. the cell harvesting, the disruption with an homogenizer. The clarification, we don't use centrifuges, but only filtrations. And then we obtain a clarified solution, and this solution, also with a concentration, is directly used for the conversion. In the last slides, we have, for instance, a picture about the biocatalysis. It's not clear because it's not enzyme, but is the solution of the substrate is not clear. Is a suspension because there is an organic part for the esters. But we use the enzymes, and at the end of the reaction, the enzyme is eliminated by denaturation. In these cases, at the beginning, the immobilization was inconvenient because the enzyme wasn't particularly stable. But now we are working to repeat the test to try to have to simplify the process by an immobilized enzymes. And in this case, however, we have improved the yield, and the wild type had a yield of transformation around 20%. Now we are around 90%.